Hi everyone, this video is about cellular respiration. As you know, cellular respiration is the breakdown of organic molecules like glucose, fats, and proteins to make ATP. We're going to focus on the pathway for using glucose for cellular respiration, but be aware that fats and proteins can be input at various steps along the way and can also be used to generate ATP. Cellular respiration is an aerobic process, meaning it occurs in the presence of oxygen. And the equation for cellular respiration is glucose plus oxygen results in carbon dioxide, water, and ATP. This is a balanced version of the equation, so you can see where all of the atoms are going. In this video, I will cover redox reactions, that's reduction and oxidation, glycolysis, link reaction, Krebs cycle, and oxidative phosphorylation. Those are the phases, the 4 phases of cellular respiration, and fermentation, which occurs when there's no oxygen present. First up is redox reactions. So cellular respiration pretty much boils down to the transfer of electrons. So that means a lot of redox reactions. The way that I remember which word means gain of electrons and which word means loss of electrons is I think about that reduce means to decrease the charge. So if the charge of a molecule is decreasing, that means electrons have to be added because electrons are negative. Some people prefer these kind of memory tricks, like Leo the lion says grr, lose electrons oxidation, gain electrons reduction. Another one you might hear is oil rig. Oxidation is loss of electrons, reduction is gain of electrons. Up to you, pick one. Might be a good way to kind of hold on to that information. In cellular respiration, glucose is oxidized. So that means electrons are leaving glucose as it becomes pyruvate and along the way through cellular respiration. And another way that we can kind of see that glucose is oxidized is if we compare glucose to carbon dioxide. We can see that glucose has a ratio of 1 carbon to 1 oxygen, whereas carbon dioxide has a ratio of 1 carbon to 2 oxygens. Now, oxidation does not always mean gaining an oxygen. So this is kind of a nice way to check it or remember it, but you don't always need to be gaining an oxygen for oxidation to be happening. A couple of examples of reduction that happens in cellular respiration, because anytime you have oxidation, there needs to be reduction. Those electrons have to go somewhere, is the formation of NADH and the formation of FADH2. So these are both molecules that can accept electrons so that they can be transferred to another location. So NADH can accept 2 electrons, and it also picks up a hydrogen ion, aka a proton, when it does this. And FAD can accept 2 electrons as well, and it picks up 2 hydrogen ions. NADH comes from NAD plus in the hydrogen and electron. FADH2 comes from FAD hydrogens and electrons. Notice 2 hydrogens are added to FAD. That's why we're calling it FADH2 versus there's 1 hydrogen and NADH. So these are the reduced form of the molecule, NADH and FADH2. Now for the phases of cellular respiration. So first up is glycolysis. Glycolysis is the breaking of glucose. Glucose break, glycolysis. And when you break the glucose, you're breaking it into 2 pyruvates. That's the name of the molecules that are created. It's occurring in the cytoplasm of the cell, so not yet in the mitochondria, outside of the mitochondria. And 2 pyruvates, 2 ATP molecules, and 2 NADH molecules are produced. This is a good time to point out that NADH is very similar to NADPH, which we talked about in photosynthesis. They both accept electrons and carry them somewhere else. I remember that NADPH goes with photosynthesis because it has a P in it, as does photosynthesis, starts with P. So here is a picture of the molecule of what's happening, glucose to pyruvate. So this glucose has 6 carbons, you can see, 6 oxygens, and it's split in half, basically making 2 pyruvate molecules. They have 3 carbon and 3 oxygen. So see how that's just kind of cut in half, and you've got two products, two of the same product on either side. The atoms are not disappearing. They're all going somewhere. They're all accounted for. What you're not seeing in the pyruvate is a couple of the hydrogens. A couple hydrogens have not disappeared, but they have been released. So if we look a little bit closer at this equation, we have C6H12O6 into 2 C3H4O3. So H4, 4 hydrogens, there's 2 of them, that's 8 hydrogens. So we haven't lost, they're not gone, 4 hydrogens. So when the electrons are coming off, when glucose is being oxidized, there are also hydrogens coming with those electrons. Some of the hydrogens are used to add on to the NADH, and some just end up in the cytoplasm. Okay, so we can see here NAD plus, and the electrons in the hydrogen, NADH, our high energy electron carrier. Next up is the link reaction. So the link reaction is a link or a connection between glycolysis and the Krebs cycle. It occurs in the intermembrane space between the outer membrane and the inner membrane in the mitochondria. So we're in the mitochondria now. It turns pyruvate into acetyl-CoA and carbon dioxide. So we can see our pyruvate molecule here, and you can see that 2 of the carbons and 1 oxygen are combined with coenzyme A, that's CoA, that's how we're abbreviating it, and that's acetyl-CoA. And 1 carbon and 2 oxygens are the carbon dioxide. So again, all of our atoms can be accounted for. The products of the link reaction are 2 acetyl-CoAs, 2 carbon dioxides, and 2 NADHs. Remember, there were 2 pyruvates to start, so they're both split. That's why we have 2 of everything. The Krebs cycle occurs in the mitochondrial matrix, so within the inner membrane. It's cyclic, meaning there's regeneration that happens, just like the Calvin cycle in photosynthesis. There is a molecule that needs to be regenerated so the cycle can continue to happen. In the Krebs cycle, acetyl-CoA is oxidized, producing 2 ATPs, 4 CO2s, 6 NADHs, and 2 FADH2s. So there's a lot of electron carriers produced in this phase. Next is oxidative phosphorylation. So this is where most of the ATP comes from. There can be about a maximum of 34 ATPs. The process is not always as efficient as possible, so we don't always get 34 ATPs out of it, but that's what's possible. Oxidative phosphorylation is 2 parts. It's an electron transport chain, and it's chemiosmosis. It takes place in the inner membrane of the mitochondria, so those kind of convoluted folds in the cristae. And these folds, or the cristae, increase the membrane surface area. So there's more physical membrane space for all of the proteins needed for oxidative phosphorylation. So you can have it happening many, many, many times, more times than if it were just a flat membrane. So this is my mitochondria. I'm going to zoom in on this part of the inner membrane right here. And you can see that I have those 2 main parts, the electron transport chain and chemiosmosis. So let's break it down. First up, NADH and FADH2 molecules come in and bring their high energy electrons. So remember all those electrons that they picked up as they were being reduced in glycolysis, link, and Krebs cycle? This is where the electrons are going. They're dropped off into the electron transport chain, and as they lose energy, as they travel through the electron energy, that energy is captured and used to pump protons from the mitochondrial matrix into the intermembrane space. So just like with the light reaction, we're building up a high concentration of protons. There's a lower concentration over here. Just like with the light reaction, those protons want to get away from each other, and ATP synthase is there for the protons to rush through. ATP synthase can capture that energy and use it to connect an ADP and a phosphate group together to make ATP, just like in the light reaction. So a couple things that are different are There are 3 proteins in the membrane here that are pumping those hydrogen ions out versus in the light reaction we had 1. Also, the final electron acceptor is different in oxidative phosphorylation. You might remember in light reaction it was NADP+. Here it's oxygen. So this is where our water product is coming from. We have oxygen and hydrogen ions, combining with those electrons that came through the electron transport chain to form water. So this is the opposite of the water splitting that we did in the light reaction. So again, we have our electrons coming down the electron transport chain. We have our oxygen. That's the oxygen we're breathing in, right? And we have our hydrogens, and those combine to form our water molecule, another product. So oxygen is the final electron acceptor in this process. So again, this was the electron transport chain, and this is the chemiosmosis. So that's the ATP synthase part. And mitochondrial matrix is on this side, and intermembrane space is on this side. I also want to point out that it's this phase that's regenerating the NAD plus and the FAD, which will go back to glycolysis, Krebs, and Link to be used to be reduced again so that more electrons can come over for this process. Sometimes there's not oxygen though, and many cells are able to do something called fermentation so they can still make ATP. So this is anaerobic, that means no oxygen present, no oxygen gas. It occurs in the cytoplasm. It's not as efficient as aerobic respiration, as regular cellular respiration. We only get 2 ATPs out versus a maximum of 38 with oxygen. That's a pretty big difference. Why fermentation is important, why we can't just do, say, glycolysis and get the ATPs out, is because we need to recycle that NAD+. That needs to be present for glycolysis to keep happening. I'll explain that a little bit more in a second. So there's 2 types of fermentation. There's alcohol fermentation, makes ethanol. Bacteria and yeast can do this, and beer and wine is made through this process. There's also lactic acid fermentation. This happens in your muscles, and that is when lactic acid is produced. So if you are exercising and you're like really sprinting, really running very hard, and your muscles start to burn, that's an accumulation of lactic acid. That means that your heart and your lungs weren't doing quite a good enough job of getting oxygen to those muscles, so they had to start doing fermentation because they ran out of oxygen. So glycolysis produces those ATPs. So why do we need the fermentation part? We can't just continue to use NAD plus until like forever, because it takes energy for our cells to make NAD plus. So they're not just going to make an infinite supply. It will run out. So eventually, if the NAD plus runs out, we can't do glycolysis anymore. We can't make ATP. Fermentation comes in by using the NADH, using the electron, as well as the pyruvate, to make the alcohol and carbon dioxide or lactic acid, but also to regenerate the NAD+, because it's using the electron, so we have our NAD+ back. That way glycolysis can happen again. So in this video, to summarize, we talked about cellular respiration. We touched on redox reactions, the 4 phases of cellular respiration, glycolysis, glucose into 2-pyruvates, Link reaction, pyruvates into acetyl-CoA and carbon dioxide, Krebs cycle, acetyl-CoA goes in, carbon dioxides and many forms of energy come out, energy molecules, and oxidative phosphorylation, that's where the big hit of ATP is coming from, electron transport chain and ATP synthase. We also touched on fermentation, which happens in the absence of oxygen. Thanks for watching.